Welcome to the Week 1 overview. This will be the Introduction to Biomanufacturing, Pathways and Model Systems. The details of this material will be provided in the assigned readings as part of this course. What is biotechnology? This is any technique that uses living organisms or components of organisms such as cells to make or modify products to improve plants or animals or to develop microorganisms for a specific use. It also encompasses all of the technology connected with plant and animal life. Biomanufacturing. Biologic organisms are also important in the production of a number of products in industry. Bacteria are especially useful in the purification and separation processes. What is biomanufacturing? This is any type of manufacturing or technology that utilizes biologic systems to produce commercially significant biomaterials or biomolecules for use in medicines, food and beverage processing, or in industrial applications. Biomanufacturing products are recovered from natural sources such as blood, or in the case of single cells, from the cultures of microbes, animal cells, or plant cells grown in specialized equipment. The cells used during the production may be naturally occurring, or they may have been modified or derived from genetic engineering techniques. Some examples of products of biomanufacturing are for medicines, things such as amino acids, biopharmaceuticals, cytokines, growth factors, monoclonal antibodies, and vaccines. Within the food and beverage industry, enzymes, and protein supplements. For industrial applications that employ cells or enzymes, things such as bioremediation or waste management techniques that involve the use of organisms to remove or neutralize pollutants. The production of detergents, as well as the production of plastics. Cellular metabolism encompasses all chemical processes occurring within a living cell. Metabolism is defined as the totality of an organism's chemical reactions arising from interactions between molecules. A metabolic pathway generally has multiple steps. It begins with a specific molecule and ends with a specific product. Each step is generally catalyzed by a specific enzyme. In the diagram shown here, we start with molecule A, which undergoes 3 enzymatic steps, ultimately leading to product D. Genetic engineering. Genes are made of DNA. These genes hold the blueprint for all aspects of a living organism. How it grows, how it looks, how it behaves, the products it makes. And genetic engineering, also termed recombinant DNA technology, refers to the alteration of one or more genes within a living organism. An organism with an altered or modified gene is termed a genetically modified organism, or GMO, which exhibits a unique genotype. There are numerous genetic modifications that are possible. Inserting a foreign gene from one species into another, producing a transgenic organism, modify an existing gene so that the product is changed, or altering gene expression such that it is translated more often or overexpressed, or such that it is not expressed at all or silenced. Genetic engineering alters the DNA code of a living organism or cell. Techniques that have currently been utilized are selective breeding, recombinant DNA, and transgenic organisms. Selective breeding breeds only those animals or plants with certain desirable traits. Selective breeding has been used for thousands of years in the context of crops and domesticated animals. And here we even show an example of a labradoodle, which is produced by selective breeding to produce a hypoallergenic animal. So an example of how recombinant DNA technology works, we show this diagram. This is the ability to combine the DNA of one organism with the DNA of another organism. You remove bacterial DNA or a plasmid. You cut this plasmid with a restriction enzyme. You then cut the DNA from your organism of choice with the same restriction enzyme. You combine the cut pieces of DNA together with another enzyme termed a ligase and insert these back into the bacteria. In the bacteria, these will reproduce and the foreign gene is expressed within that bacteria. And when these are grown in large cultures, the particular product of that gene is then released into the media where the protein of interest can. Here are some examples of the benefits of recombinant bacteria. A gene for human insulin or human growth hormone can be inserted and grown in bacteria. Or bacteria have been engineered to eat oil spills. Not only can the DNA of bacteria be altered, but the DNA of plants may also be altered. And the result of these can be disease-resistant or insect-resistant crops, hardier fruit so they can resist a variety range of temperatures or of need for water. And though we may not realize it, up to 75% of the food in most supermarkets is genetically modified. Genetically altered crops and animals are among the most significant applications of bioengineering. For example, a farmer is able to use less herbicide if the plant has been modified to be weed resistant. In this slide, we show a simplistic diagram of how you would create a genetically modified plant. First, you create a recombinant bacteria which has the desired gene to produce the characteristic you're looking for: weed resistant, drought resistant, insect resistant, whatever the desired effect is. You allow this modified bacteria to infect the plant cells. Then this gene from the bacteria has now been inserted into the plant chromosomes, where it is then carried or perpetuated within the plant, and the plant develops the characteristics of that gene. Again, as I indicated, we may not realize it, but most of the food, or many of the foods that we come in contact with, have at least some component of genetic modification. For example, 50% of all of the cotton has been genetically modified. Much of the sugar beets, where about 50% of our sugar is derived, has been genetically modified. About 87% of the canola oil has been genetically modified. So, without realizing it, we come in contact with genetically modified foods every day. So not only can we modify bacteria, plants, we can also modify or genetically modify animals. this produces transgenic animals. These, for example, could be mice. They're transfected with or have the gene for different components of the human immune system inserted, enabling us to study in an animal model various components of the human immune response. Certain genes can be inserted into chickens to make them more resistant to infections, so that we have to use less antibiotics during chicken growth. And cows can also be transfected with genes to increase milk supply or leaner meat. Then there are other livestock, goats, sheep, pigs, which can have human genes inserted so that they produce human proteins and secrete them into their milk. So cells can serve as a factory. Cells function as a live chemical factory with a specific input and a specific output. In this, we show a stimulus that elicits reaction A, which then goes to reaction B, and on the normal pathway leads to reaction C in a specific product. But in the genetically modified version, reaction B, instead of going to reaction D, now goes to reaction C and produces altered product. An example of this would be using a pig-derived pancreatic cell and inserting the gene for human insulin, so that when the cells are stimulated, instead of producing porcine insulin, it now will produce human insulin. Again, here is a separate example of where E. coli is used to produce human growth factor. In the same way we discussed earlier, a bacterial plasmid is isolated, it is cut with a restriction enzyme, and then the human gene for growth hormone is then inserted into that plasmid. The material is now treated with a ligase. It now has back to its intact circular form of the plasmid, which is inserted into the bacteria, where it is then grown in large quantities in a bioreactor, and the human growth hormone is released into the media of these cells, where it can then be isolated and processed as a medication. Model systems. What are some of the fundamental issues for bioengineering? The complete genomic sequences are not always available for all organisms, and even when they are available, the functions and interactions of only a small percent of the genetic components are clearly understood. For example, within humans, only about 5% of the genes have known functions. In contrast, within E. coli, more than 60% of the genes have known functions. So by understanding gene functions, we are able to predict, control, and modify their responses to specific manipulations. So what is a model organism? Many aspects of biology are similar between most or all organisms. There is a preservation of many gene functions across multiple species, from microbes all the way to humans. And it is easier to investigate specific traits in a particular organism. For example, genetics are easier to study in small organisms that breed quickly, and these would be very difficult to do in humans. The most popular model organisms have strong advantages for experimental research. And many of these organisms have been investigated for many years, where techniques, genes, and other critical information have previously been discovered, and that information has been disseminated among investigators. So in models, it's the data that represents the product, with an accurate representation of the process and equipment as the goal. While in most cases the ideal or ultimate target system are humans, this target system and the industrial equipment needed to provide the optimal data can be very time-consuming and expensive. As a result, models are used because these models are easier to use and we have a better understanding of them. The problem is that these model systems are a compromise. The shorter time to get the data and the less expense results in having imperfect. But the importance of a model system, it allows you to go from a high quantity to a high quality commercial applications. When you initially begin your study and you have a number of genetic modifications under a number of conditions, you have a high quantity, can be hundreds to thousands of different conditions that you want to investigate. This can be achieved in microtiter plates, where you can look at high quantity or high throughput conditions. Once you narrow these down to a few dozen, 10 to 20 conditions, you then could do these in a slightly larger situation where you're using a culture flask, because your ultimate goal is to grow these in large batches in a bioreactor where liters of material containing your desired protein product can be derived. What are some of the available models? Many well-characterized model systems are available. Models can be single or multi-celled organisms. Some of the commonly used examples are bacteria, viruses, yeast, insects, mice, rats, hamsters, zebrafish, chickens, slime mold, plants. These models have been investigated for many years, and their characteristics and information are well known and extensively distributed. Here's a list of some of the commonly used organisms. Like I said, bacteria such as the E. coli, yeast, plants, fruit flies, mice, and generally the ultimate target organisms for these models are Homo sapiens, or man. An example of why would you use E. coli as a model organism? Well, it has a high growth rate. It's genetically amenable. Its metabolism is well understood. Its natural production of organic acids, its ability to grow in various substrates, its plasticity of its metabolism, and it has high theoretic yields. And here again is an example of some of the organisms used for metabolic engineering. Things where bacteria can be used for organic acids and biofuels, bioethanol. Some can be used for therapeutics and enzymes. Some bacteria can be used for removal of waste products. Some can be used for antibiotics or recombinant human proteins, as well as production of gases. So what are the limitations of these models? Like I said, the ultimate target is generally humans. And it's important to recognize the biology of humans is unique in many instances. Humans are not simply mice without fur and a tail. Regardless of the detailed understanding of other animal systems, in some cases there is no alternative but to study humans. In those cases such as cancer, pregnancy, and certain genetic disorders. The human genome has now been completely sequenced. However, the functions of many of these genes remain unclear. And additionally, beyond simple genetic differences, humans exhibit unique protein post-translational modifications that are critical mediators of disease and cannot be duplicated in other systems. So in summary, using biologic systems, biomanufacturing can produce commercially significant biomaterials and molecules. Biomanufacturing is mediated by manipulation of cellular metabolism, which encompasses all chemical processes occurring within a living cell. This manipulation can be created by genetic engineering, which alters one or more genes within a living organism. Thus, genetic engineering of model organisms is a key player leading to biomanufacturing and defining the ultimate conditions for production of these biologically significant molecules.